So the next speaker is David Hartlmayr from Selenium Proteomics. David is application chemist at Proteomics and Selenium. And I guess we talk about the platforms and solution that Selenium has to actually bring the single cell into a well without losing cells.
Thank you all for coming to today's Single-Cell Proteomics Day. And my name is David Hartlmayr. I'm application specialist at Selenium. And today I will introduce you to the Cell1, which is the instrument used for all our workflows. So the Cell1 basically is a nanoliter pipetting robot that's capable of cell sorting while also being humidity and temperature controlled. And all these features are quite interesting in the field of single-cell proteomics because you really want to go as low as possible with your reaction volumes. First, to reduce your peptide losses in a workflow. And second, because you want also to reduce your background chemicals for the mass spectrometry analysis. So dispensing with the Cell1 works is acoustic-based. So you can see here this glass capillary. And second. This is the glass capillary. And here it is surrounded by a piezoelectric ceramic. And here a certain voltage is applied. And this voltage induces a droplet to be ejected at the tip of the vessel. And with this, we get really reproducible droplets of around 300 picoliters, but of course, this highly depends on the parameters like the voltage and pulse, and also on the capillary diameter. And we dispense droplets at around 500 Hertz. And so the solution, it can be aqueous, it can be organic, is aspirated into the nozzle and can then be dispensed based on this acoustic technology. The image-based single-cell isolation works by aspirating the cell solution of around 200 cells per microliter into the nozzle and placing it in front of this camera here. And here the cells are imaged. And then when there is only one cell at the tip of the PDC, so the capillary, then a single cell is dispensed at the target. This way we can isolate cells at around 40 cells per minute and with high accuracy. And since we only work in really small volumes, it's actually quite perfect for rare cell species where there are only a few microliters of sample there. So we don't need milliliters of sample to do our single cell isolation. So now in a little bit more detail, so the cell suspension is loaded into the glass capillary. Then the capillary tip is, as I said, placed in front of the camera. And then in the processing of the software, the tip is segmented into two zones. The ejection zone, which represents the volume of the next droplet that will be produced, and a safety zone, which is really here to guarantee that there is no second cell in this droplet. And all cells that are dispensed are actually imaged, and the image is saved, and all parameters like diameter and elongation are saved on the Cell1. So this can be used also afterwards for data analysis. Actually see here the parameters that can be used for the cell isolation. So we can use min and max diameter and also elongation in a minimum or maximum way. And in addition to that, we also have 4 fluorescence channels that can be used for cell isolation. And like this, you set all the parameters, and then the cell isolation can start. And you can see here on the right, there's a single cell that will be isolated to the target. And here is no single cell. So this will be dispensed to the recovery vial just below the camera. And here is a short video. You can see here the machine is sorting in live speed, and in the bottom video you see the capillary tilted 90 degrees, and all the single cells are isolated to the target, but here the doublet is just dispensed on the recovery vial just here below. And like this, as I said, around 40 cells per minute can be isolated. And yes, so with this technology, we are able to isolate with bright field imaging cells of many different ranges of sizes, shapes, and also with fluorescence. So with bright field, we are in the range of 3 micrometers up to 80 micrometers. And with fluorescence, we can go even lower to 0.5 micrometers. You can actually also combine both technologies for your cell isolation. But then, of course, the cell isolation will take a little bit longer to accumulate all the information for the cell isolation. So, and in 2019, I was still working in Karl's lab together with Claudia Zateska, who was a PhD student there. And then the collaboration with Selenium started, and we developed this Proteochip protocol. And it's actually quite similar to a standard TMT protocol, but just in much lower volume. And it's all the standard steps that are used in proteomics are also used here. And afterwards, we measured with the Thermo Fisher Orbitrap Explorers 480 with the U3000 as a HPLC. And I will just show you quickly how the chip is made up. So it's each chip contains 12 arrays, and each array contains 16 wells, and every well is already preloaded with an oil to avoid all evaporation, because you can imagine when you work with nanoliters of volume, evaporation plays a huge role, which is also the reason why we work with high humidity. And up to 3 chips can be processed per run. And like this, when you really use every well for a single cell, process up to 476 cells per run. In the first step, we dispense our master mix, which already contains a protease, the buffer, and lysis detergent into each well. And this is followed by cell isolation based on the criteria that are set by the author. And this is followed then by 2 hours of 50-degree incubation at high humidity. And this is followed by TMT labeling of the respective wells, another 1 hour of incubation of the TMT label, and afterwards the samples are quenched. The pooling step. Pooling, it is really easy. There's no manual step involved. It's quite easily done via centrifugation. Within 2 minutes, the samples are pooled, and up to 6 sets of samples are ready to be analyzed. So the samples are pooled into the top part of the protochip, the so-called funnels, and those contain then the 36 samples, and this can be then also placed inside of a holder, and this holder can then be also placed in the outer sampler, and so the samples can be directly injected. And with this, we have the complete multiplexed workflow, and there is no manual steps needed in this workflow. And like this, you really can boost the reproducibility of the experiment. And yeah, this is what we, together with Karl's lab, developed. And we also did then some experiments with HeLa cells, and we looked at different carriers. So the 100x carrier, 20x carrier, no carrier samples were acquired then. And here you can really see that you have to make, you have to choose kind of a little bit if you go more in the direction of analysis depth or in the direction of high quantitative accuracy. So you can see that the 100x carrier obviously has the most IDs, but when you go in the really low carrier amount or no carrier, then the signal to noise ratios just increase. But this is just a trade-off that you have to think of, experiment to another experiment. Do you need a carrier? Are the cells big or are they small? And I think Erin already told quite a lot about using the carrier correctly. So there's a lot of nice publications out there that focus on that in more depth. And as a next experiment, we did HeLa hex cells within the TMT set, and we analyzed 170 cells like that. And we were very happy to see that we could also cluster those two cell types based on 1,900 peptides, and that we also could see respective proteome characteristics in our proteins that were identified. And then we also got some very nice cardioids. They are in vitro derived cardioids by the Mendian lab at IMBA in Vienna, and those are self-organizing into these chamber-like structures and mimic the early cardiac development, and where you could not resolve these differences of the two cell types that are predominantly there. In single-cell proteomics, you actually can do this. So we were successfully seeing the co-differentiation into early cardiomyocyte and endothelial cells based on around 1,100 peptides of the 150 cells. As a next step, we also introduced the label-free workflow, and here we worked really closely together with Brucker also. So this workflow is a little bit simpler than the TMT workflow, just because there is not the TMT step after that. And then it was analyzed with the TeamStuff SAP. And these were our initial results. So we analyzed 1, 5, 10, or 20 cells. And I have to add here in these results, I mean, the samples were lost somewhere in Paris for 1 or 2 weeks, and there was no dry ice anymore. So we were really happy to see that we still got around 1,000 proteins per cell, so per label-free cell, and that indeed, if you increase the number of cells, that you also increase your analysis depth. Christoph from Brucker also looked a little bit deep into the data, and he saw that you could really nicely distinguish also the two proteomes of these cell types. You could first cluster based on the PCA plot, and you could also see, okay, the protein abundances are also quite different, and also some specific proteins for each cell types could be identified. Then also we worked together with Evosep, and in combination with the Whisper40 SPD method and the TeamStuff SAP, we generated some data. And we were very happy to see that we could then, with this 40 SPD method and very nice acquisition methods by Brucker, see that we got around 2,000 protein groups per HeLa cell without match between runs. And when you look at the injection of 250 picogram of HeLa bulk digest, you can see that we are actually quite close to what is injected there. And this was also quite nice to see. And Dorte from Evosep also looked a little bit deep into the data, and she could show that when you compare the single cell data to bulk digest that's diluted to also nanogram or picogram ranges, then you can see that actually some regions are quite differentially expressed, and very nicely you can also see that the single cells here, this represents quite nicely the heterogeneity even in HeLa cells. So this motivated us also that, okay, with single cell proteomics, you could also investigate this heterogeneity in these single cells. And with that, I would really like to thank all our wonderful collaborators, Brooke, Timo Fischer, and also in Karl's lab, and Claudia. Without all these wonderful scientists, all this wouldn't have been possible. So with that, I thank you all for listening. And if there are any questions, please let me know.
Thank you very much for your talk. Very, very interesting. Again, questions? Okay. Oh, sorry.
Thank you, David, for your talk. And a very general question. Speaking of source of cells, so we have seen cell lysate. How far is this workflow for tissue?
You mean like with laser microdissection
Yeah,
methods?
Well, yeah.
Yeah.
If I'm a scientist, I'm asking you, okay, I have cell lysate and I have tissue.
Mhm.
What would you suggest to proceed with, and how far this workflow can be tackle the tissue?
So for now, what we have until now analyzed are cell lysates that are just dispensed in really low volumes, and this has worked actually quite nicely for us. And we are working currently also on solutions to integrate like the Salmon with the laser microdissection. So this is all work in progress right now, but I'm sure that there will be also some workflows coming up.
Yeah, I was also thinking about, I mean, in line with what Barbara just mentioned, how do you deal with those cell models that basically only grow in adherent conditions that basically form an epithelium, for example? And as far as I know, but I'm a chemist, I mean, you need to be very fast in treating those cells when you basically take them out of their environment, which is an epithelium, and you do whatever omics you want to do. How do you deal with this?
So usually we directly before the experiment, we use trypsin to remove the cells from adhesion. And after that, we directly wash them with PBS and aspirate it into the capillary. And when you're really quick, I guess the proteome changes should be still within the range that the proteome can still be differentiated, right? So, but we have also thought about fixation methods that could be maybe used, but we haven't done too many experiments with that. But yeah, I think this could be solving this kind of to get a fixation method in the proteomics.
Other questions? No? Questions?
If we still have time for a very quick question, can I? Maybe it's a silly one, but I understand that with the CELN1, you basically can do also like fax sorting, right? Maybe it's not designed for that. Is it like? I mean, I understand all the advantages. Like, what would be the disadvantage compared to using it as a fax sorter? Is it like slower?
I mean, what's installed at the instrument right now are LEDs and not lasers.
Okay.
So that's the main difference, I would say. So the fluorescence signals or the visuals that we get are not as strong as with fax sorting. So this would be one of the points, but we, on the other hand, can really image the cells. So I would say the big plus point for the cell one is that you get afterwards really the image of each cell, and you can analyze it like that. And we can only use the 4 fluorescence channels that are introduced. So together with that, you can do some fluorescence sorting. But what's actually could also be quite useful is to previously fax sort the cell populations and then really dispense with the CELN1 to each well.
Exactly. Thank you.